And thank you so much for that nice introduction. I won't go into too much depth here because of that, but yes, I am studying oceanography and astrobiology at the University of Washington. My specialization in both is in molecular microbiology and specifically bacterial, archaeal, and viral metagenomics. Maybe metagenomics is something we're familiar with here. I would imagine probably genomics is something we're familiar with. So what I hope today is to explore these methods, the limitations that we encounter with these methods in an applied sense, and also what the current, not only these methods, but what our current understandings are in terms of the interactions of oceans and climate, and also climate and human health. So quickly to define this term that we're seeing, metagenomics, many of us are probably familiar with genomics, where we're looking at the genes in a genome for a particular organism. The meta in metagenomics essentially comes in where we start looking at the genes on the genomes of all of the organisms in a sample or a mixed environment or a mixed community. And as you can imagine, that there are then tricky ways with which we can piece apart the individual genomes of these individual organisms in these mixed communities, and we'll get into that in methods in a second. So that's more of what I do. What's really driving this talk more is this question: why should we think about the ocean, and really expanding that to the environmental system in the context of human health and planetary health? Many of us might be wondering why we're talking about the ocean in a biotech space. Mostly and probably because when we think about the ocean, this might be what we think of. We know these charismatic megafauna, as us biased microbiologists refer to them, whales, fish, these things that a lot of people gravitate to when they think about the ocean. Believe it or not, it's a question we get a lot in oceanography. We don't actually study any of these. We start looking at some of the larger scale diversity as you start to expand this classical definition of what we see in the ocean. So as we start to add nuance to this plot, we can see more organisms doing more things, more on the microbial level. And importantly, when we introduce this lowest level, this lowest end, the smallest size spectrum in the marine environment, we see we have bacteria, we have viruses, along with that we have archaea, we have single-celled eukaryotes as well. And these organisms are doing a lot of incredibly important things in the ocean with relevance to the entire planet. Indeed, when we look at the distribution, I'm sorry, it's a little glitchy. When we look at the distribution of these organisms, we actually see that over 70% of the ocean's biomass takes the form of bacteria and protists, or single-celled eukaryotes. This is important because these small guys that often go overlooked because of their size are actually doing some really significant work. Or performing really significant cycling in the marine environment. Indeed, if we look at this picture here, we can actually see the abundance. This is just an image of seawater, and we can see large dots. This is an epifluorescence microscopy slide for those who are familiar. Large dots which represent cells, and small dots which represent viral-like particles. You can see their abundance and how numerous they are in the environment. So they're numerous, and therefore there's a lot of them. They're containing a lot of carbon in the marine system. And so what are they doing? When we take this and we apply it to our understanding of, let's say, the marine food web, we see that we have our important charismatic megafauna that are engaged in more of a standard trophic interaction or trophic ladder, something that we may have learned about in normal biology classes, where we have a transport of carbon from foundational autotrophy up into more complex organisms. The more nuanced side of the marine food web, at least, is what we would define as the microbial loop here on the left side of the screen. In this microbial loop, these are composed of those, what we now know as the dominant biomass in the marine environment, these small organisms that are constantly cycling nutrients both in and out of cellular material, transferring that up trophic levels, exporting that material to depth, and importantly, interacting with inorganic species, like, as you see at the very top, carbon dioxide, which we've heard so much about in the context of climate change. Indeed, when we look at our planetary system in the context of this key molecule, carbon dioxide, we oftentimes think about the atmosphere, which makes sense. That is where a big reservoir of carbon is. We can see about 730 gigatons of carbon is contained in the atmosphere. In addition to that, about 1,500 plus 500 gigatons of carbon are also contained terrestrially in soils and plants on land. But importantly, and what people tend not to realize, is that when we look at the oceanographic reservoir, the ocean reservoir, that's 38,000 gigatons of carbon contained in the ocean. And this carbon is a dynamic reservoir that's constantly being cycled, mostly by this important biomass, and also through inorganic cycling. Looking at the ocean cycling specifically, in a second, we see it's rather complicated. There are a lot of both acid-base reactions, there's uptake, there's cell lysis, there's reintroduction, there's death and decomposition, all occurring here. And so without getting too specific in here, we can simplify this view by looking at our major planetary layers. So we have atmosphere, surface ocean, deep ocean, and seafloor. So we know one of the big problems is that we're getting an increase in carbon dioxide in our atmosphere. Even our naturally occurring carbon dioxide in our atmosphere constitutes a large percentage of the air. So this carbon dioxide naturally diffuses into the water. And this is probably something that we've seen in science courses, where this carbon dioxide engages in acid-base reactions to reconstitute its speciation. This acid-base reaction produces notably bicarbonate and carbonate, which are actually the dominant forms of that become integrated into cellular biomass. And importantly, when that happens, this allows for an accumulation of this carbon, sinking of this carbon either through cell mortality or the normal circulation of the ocean. And as you can see, we're getting farther away from our atmosphere, or from this problematic reservoir in the context of climate change. Ideally, if this sequestration occurs efficiently, it can reach the seafloor, where we really call it sequestered or removed from the atmosphere for at least relatively long timescales. So we saw from this that there are three main sinks for this carbon system: the soils, the plants, and the oceans. And importantly, looking specifically at our climate change-driven enrichment of atmospheric carbon dioxide, the oceans have already absorbed 30% of our total emissions from the start of the Industrial Revolution. So they act as a very important sink for carbon in our planetary system. So we've talked now about one interaction by which the ocean and its organisms, its life, impact climate and impact our planet. I wanted to take a moment here to talk about how we actually study these things. And I wanted to use an example of specific work that I've done recently, studying metagenomics in deep sea volcanoes or hydrothermal vent systems. I think this is an interesting example that hopefully will illuminate some of the limitations we're operating under doing applied work in such extreme environments. So first, here is an image of this of these extreme environments. Sorry, let me move something quickly. These are hydrothermal vent systems imaged from the deep sea off of the Washington coast. These systems are magmatically charged, so subsurface magma heats up water that's been entrained into the seafloor and chemically alters it, creating incredibly unique biochemical and biological environments. Indeed, these systems can help support primary productivity or the fixation of carbon in the complete absence of sunlight, and it's thought that life in these environments might mirror a lot of the life that we might expect to see, for example, on other planets like icy moons. It's also thought that these really unique biochemical environments are relevant for example, the origin of life, and I will get back to that later. Studying these environments are difficult. We're interacting with 400 centigrade water at pressures many, many hundred times that of atmospheric pressure, oftentimes operating remote devices to do our sampling. Even just getting there sometimes can be a limitation. In this case, In this case, I was sampling from a hydrothermal vent system located about 350 nautical miles off of the Washington-Oregon coast. In order to get there, here's a map of that vent field, targeting an ASHES vent field and International District vent field is what we call them. In order to get there, we have to use relatively fancy equipment. This is a partnership between the United States Navy and various universities across the United States to operate this fleet of oceanographic research vessels, or essentially floating laboratories that we can take to anywhere in the globe with the right funding and sample what we want. This is actually a picture of the Thomas G. Thompson. This is University of Washington's vessel. And I was on this about a year ago, a little bit over a year ago, with an excellent team of microbiologists, ocean technologists, engineers, submersible pilots, astrobiologists, geobiologists. It's an interesting team that's selected for each specific research question that we're trying to address on these expeditions. On this vessel, sampling started with the deployment of a remotely operated vehicle. I've put kind of a human size comparison on here so you can get a sense of the scale of these devices. We send it over the side and we deploy it sometimes 2, 3, up to sometimes 5 kilometers below the seafloor. This is so far that it's thought normally that you're steering this device around a ship, but really there's so much tether deployed that the ship is actually pivoting around the deployed tether when you have a 3-kilometer tether deployed. We then go into this, what you might think of as a mission control, to remotely operate the vehicle. We have a line of engineers, pilots, and navigators in the front, and the scientists sit in the back to help with the operation of the vehicle to pursue our science goals for this deployment. We saw some videos of this device collecting seawater, for example, at these deep sea vents, having to fend off pressure, darkness, cold temperatures, corrosion, and a whole slew of other challenges. After deployments like these, we tend to also deploy some other sampling equipment. One of the key or classic sampling tools for oceanography is called this CTD rosette. CTD stands for conductivity, temperature, and density, and it represents the key sensor arrays that we have mounted on the base of these rosettes. And importantly, these big gray bottles act as bottles or buckets that we can fire at any depth and collect water when we see the appropriate signal. And these signals can be chemical, they can be physical, they can be biological, based on the sensor output we're getting from the base of the rosette. These we deploy also down to many kilometers below the surface. Here's a picture of one of those deployments being craned over the side. And once it's deployed, we then go to the ship's computer lab, separate from the mission control, where we conduct sampling. For example, in my recent sampling, this included what was called a plume hunt. So I was looking specifically for those volcanic plumes emitting off of the sea. To do this, you look at the temperature the sensor output, in my case, temperature and light scatter, to look for key signals or anomalies that would be indicative of the right water parcel type that we're targeting. This involves corresponding with a winch that operates the profiling of the sensors, looking at your screens to evaluate the chemical conditions that you're targeting, and then also simultaneously communicating with the bridge of the ship to have it make strategic translations to drag the sensor and essentially profile the deep sea 3 kilometers below you for the right signal, which, as you can imagine, can be challenging in a vast ocean. Then, upon sampling, one of the other natures of the applied sciences is that we're often limited by factors out of our control. For example, the ocean is an extremely low biomass environment, meaning that while I mentioned that there's a lot of biomass when you integrate the entire volume of the ocean, for any specific parcel, you're often limited by what you can collect. And so for my case, I had to collect 75 liters worth of plume water, and oftentimes I'm limited by what's feasible to collect on these cruises. And then I have to go through concentration steps to make the sample usable. On the ship, for example, this involved passing my sample through what's called a transverse flow filter or a tangential flow filter, which is imaged here. But to visualize what it's actually doing, we can see that we have our input water, in this case about 75 liters, that goes into this TFF. The flow of the TFF crosses tangentially across the filter membrane, going back into our raw water sample. This flow also allows water to be removed from the sample. It essentially only allows water to be removed from the sample, and it retains biomass in the form of cells or viruses on the input side. With cycles, what this generates is your input sample that has all of its water removed. And so I'm able to take 75 liters worth of this volcanic plume biomass and concentrate it down to about 150 to 200 milliliters of sample, which is something that I can work with back in lab. I then, on the ship, cryopreserve these samples, oftentimes with liquid nitrogen or with deep freezing, to be able to transport them back in a way that doesn't also harm the samples. And what I've just described, this process alone took 32 hours of continuous shipboard work to collect this one sample. So you can see the limitations we're operating under, let alone the seasickness and whatever else we might be experiencing while out in the middle of the ocean. So once we're back in lab, I performed a high molecular weight DNA extraction on these samples. Oftentimes these extractions are not following a standard protocol released by whoever we might use for these extractions. Oftentimes, because of the tricky nature of our samples, we have to use sort of hybridized protocols that take into account, for example, our biomass limitations, or take into account the nature of our community structure that we're sequencing, or take into account, for example, the harsh contaminants that might just be present with our sample at the time of sampling. After we go through this process, we then usually sequence. In this case, I was using Oxford Nanopore Next Generation Sequencer to produce a high-quality sequence set that might be something you're all quite familiar with. Indeed, here's just a screenshot of one of those FASTQ files. So after this, you might now be wondering, okay, well, we have sequences that come from a bunch of different organisms. How do we actually go and piece apart information on any one specific organism? Well, that can be complicated. This is just a simplified workflow of my recent workflow, where I was targeting host-virus interactions in these samples. I don't want to focus on all of this. I want you to see that it can get crazy. The key step, though, in metagenomics that I want to focus on is what's called binning. In this process, you can see a visualization here where we have a mixed community represented by these circles. These are circular genomes from our sampling. These are spliced up and sequenced in fragments, which are these sequence reads, or that screenshot I showed you a couple pages ago. We can assemble those into contigs using relatively standard pipelines like other genomics workflows might use. And this is where we now have a challenge. We have a mix of contiguous sequences from many, many thousands of organisms that we sampled in an environment. How do we reconstruct their initial genome? Well, in binning, what you can use are genetic features. That could be, for example, tetranucleotide frequencies. That could be methylation patterns. That could be coverage of our actual sequencing, to essentially look for clustering or shared behavior between all of our sequences. You can see some of these clusters visualized here on one of these binning outputs. These clusters now form our hypothetical genomes, our reassembled genomes, which are called metagenomically assembled genomes. And those are often what we work with. Indeed, here is just a standard figure, an output from some of my recent work, which looks at bin completeness, which we have modes of evaluating, taxonomy identifications of these bins, and you can see where these bin populations fall, some better than others, some more complete than others. If we look at what each of these bins might actually look like from a sequence standpoint, we see something more like this, where each of these groups might be regions or might be behaviors of genetic information that have been assigned to a specific bin and later taxonomically characterized to a specific organism. Okay, so that's just a little dive into how we actually go about studying these environments. Now I want to take a look back and say, okay, we know how the oceans interact with the planet, at least one way in which they interact with the planet. We now understand some methods and limitations by which we can study them. Now let's look at how what those interactions mean and some other interactions between oceans, climate, and human health. So we kind of talked about global photosynthesis and carbon drawdown or carbon cycling. It's very relevant to climate change and carbon emissions. Next, let's talk about nutrient regeneration, nutrient cycling, and global biogeochemistry. So going back to this classical view of this food web, what these arrows really represent is the cycling of nutrients. Dominantly here, we can see carbon as a key cycle, especially in this carbon fixation step at the start, or at the top, going into autotrophic organisms. But we can think of this as a whole web of nutrient cycling, nutrient regeneration. One key example of that is, for example, on our microbial loop, this viral shunt, where viral interactions with these microbial species can lyse the cells through normal lytic replication and can release organic matter, not just carbon, all of those nutrients, back into the environment, into what we would call this DOM, or dissolved organic matter pool. That's one example of how nutrients are constantly being recycled in this global environment. Well, what happens to this in the context of climate change? It's estimated that with warming conditions, we'll see an increase in what's called stratification. And that is essentially just the layering of water parcels in the ocean, oftentimes going from warmer to colder, or less dense to more dense. In addition to that, this stratification, this density, is related to the cycling of ocean systems and the transport of nutrients into, for example, the surface ocean. So this image is showing, with climate change, increased stratification in the surface ocean and increased nutrient input through what's called upwelling of deep nutrients into the surface ocean, which stimulates surface primary productivity photosynthesis. Okay, well, this might sound great. We have more primary productivity, we have more carbon drawdown. What's the downside to this? Well, first, an increase in nutrient load, an increase in cell densities and viral densities and lysis has been shown to also impact, of course, increased resources for vectors that can carry diseases, as well as increased resources for pathogens themselves, as well as increased genetic density for gene transfer and potentially the increase of virulence factors for some of these pathogens. Also, the other side, the biogeochemical side of this story, is that when you have an increase in stratification and an increase in primary productivity, you also have a decrease of nutrient transfer between different regions of the ocean. One of these, let's maybe call it a nutrient, is oxygen, for example. And when you have an increase in stratification in the surface ocean, you have a decrease in the amount of oxygen that can reach your deeper oceanographic levels, or your depth. This here is a map of oxygen deficiency in the ocean. And these oxygen deficient regions are zones in the ocean where our dissolved oxygen levels essentially reach zero, which makes, as you can imagine, life via respiration at least quite challenging. Unfortunately, with an increase in stratification, we're expecting to see an increase in these oxygen deficient zones. Even more unfortunately, these oxygen deficient zones, First, here is a visualization of what that might look like. You can see just how expansive these zones are, compared especially to, for example, the North and South American continents. Unfortunately, many of these regions also correspond with our key fishing regions, for example, off the coast of South America, Chile and Peru. These fishing regions are globally relevant for both socioeconomy and for nutrition. And with the expansion of these oxygen deficient zones, we see potential impacts in fishing yields. It's thought that 10% of the global population relies on fisheries for their livelihood. This is predominantly in the global south, which also accounts for 50% of our global catches. And over 3 billion people rely on fish as their primary source of protein. This extends beyond even just challenges to fisheries, though, with elevated carbon dioxide levels due to disruptions in this ocean atmosphere cycling We also expect to see decreases in key micronutrients for all of our crop yields terrestrially as well. This is a figure showing in green foliage crop and in yellow wheat crop, and their expectation with enriched carbon dioxide levels in the atmosphere. This trend also holds true for amino acids and proteins in our food. Once again, the ocean might have a solution to this, which is the use of ocean plants for nutrition. Something that I think maybe some of us here are thinking about is the use of algae or algae-based products for things like nutrition, for example. It is thought that, for example, in this case, Palmaria palmata, which is a type of kelp, outmatches our crop counterparts when it comes to key nutrients. And so many people are also thinking of this as an intervention to both address the micronutrient issue, but also address carbon sequestration, where if we can grow this carbon, or if we can grow these autotrophs, we can sequester this carbon in the form of their biomass, and then we can ingest it. And so let's think about what this actually means in the context of sequestration. Well, so this is the system with which we've already seen before. And if we add humans and land to this system, we can imagine what would happen if we diverted some of this carbon to us. It is probably true that ingestion of this carbon would lead to sequestration to some degree in the form of biomass growth by us or by whoever's eating these kelp. It's also true that in metabolizing this ingested carbon, we're also respiring it right back out into the atmosphere. And so really what this introduces is a key concept when we're evaluating our interventions in the context of climate, which is residence time. Yes, it's true that this probably would have an impact, a beneficial impact. But also we have to think about on what timescales that impact is operating. So for example, the atmosphere, interventions in the atmosphere, they tend to cycle on the order of months. In sort of terrestrial interventions, or let's say anthropogenic interventions, those might be on the orders of 10 to hundreds of years. The minute we start to export carbon into the deep sea through, for example, cycling like I showed earlier, now we're talking on the order of thousands of years, which is the amount of time it takes an average water parcel to cycle through the deep sea. And the minute we can get that sequestration to the seafloor to be truly sequestered, now we're talking interventions on the order of geologic time. Okay, so that's kind of an entry into global biogeochemistry. At this point in the talk, we've talked about a good number of environmental cycling issues. There's one more that deserves attention, and that is extreme weather events, which I think we're all quite familiar with, especially recently. So when we think extreme weather events, I think often we think atmosphere, which is fair. That is where a lot of these weather events occur. But when we start to think about what might be driving these extreme weather events, the reasons behind these atmospheric anomalies, we again start to see trends in the ocean, or indications back to the ocean. One key trend would be probably something we're all familiar with here, the El Niño-La Niña cycle, or the El Niño Southern Oscillation. This is essentially an ocean phenomenon where atmospheric wind patterns, the reasons behind why these atmospheric wind patterns change, we actually aren't too sure about. But there's a change in atmospheric wind patterns which causes warming or cooling of surface ocean in the South Pacific. This corresponds with a shoaling or a deepening of essentially this deep, cold thermocline. You can see these anomalies for a La Niña season on the top and an El Niño bottom, where we have cold and warm anomalies, respectively. This change in sea surface temperature in the South Pacific has global climate issues or impacts. We might know many of them locally. For example, during a typical El Niño winter, we might expect to see a lot of rain, heavy rain. This might be something we actually remember from this past year here in Southern California. During a La Niña cycle, we might also, for winter at least, we might expect drier conditions. We know that quite well with drought conditions here in Southern California. But these conditions aren't only affecting North America, they affect the entire globe with extreme weather events, whether that's in the form of storms, in the form of drought, in the form of extreme precipitation, or also harmfully changes in weather pattern from whatever our historical normal is across many parts of the globe. Indeed, for 2023, it was predicted back in 2022 that the oncoming El Nino season would lead to some of the warmest weather we had ever seen, predicted potentially that we would even surpass our 1.5 degrees C threshold for global warming. And indeed, we saw that 2023 at the time was the hottest year on record, hitting a 1.48 degrees C average global temperature increase. And it's thought that these systems will only increase in frequency and increase in severity or intensity. So when we think about what impact to health these extreme weather events might cause, well, there are some obvious ones. For example, warm conditions, we might have dehydration and heat stroke. There's wildfires that can lead to displacement, but can also lead to airborne particulate matter, causing respiratory irritation and illness. In cold conditions, we can see infrastructure failure, especially, for example, like let's say hospitals and power supply. There's flooding, displacement, and importantly, the mental health impacts of, for example, being displaced from your home or having long-term mold damage from flooding conditions. And of course, watershed contamination, which can impact a whole slew of issues, including infectious disease. Can we think of any more that might be less obvious? Perhaps that might be changes to regional agricultural potential and impacts to what food we can produce where and what food we can get where. And also decreased carbon fixation efficiency. With increased temperatures, we see lower efficiency of photosynthesis and increased activity or rate of respiration, leading to challenges in crop yields and nutrition. Let's a little dip into extreme weather events. To round out the presentation, I want to talk a bit more now in this topic of genetic diversity, which relates to our last couple slides. As I alluded to earlier, the ocean is an incredible reservoir of genetic diversity at all size scales, but especially at the microbial level. When we were looking at these images of hydrothermal vent systems, I mentioned that there's thought that these sites may have actually been the site of the origin of life. Indeed, with the discovery of one particular kind of hydrothermal vent field, which has a specific type of bio and geochemistry occurring there due to essentially the structure of the rocks beneath it, it was found that this might be a really probable site for the origin of life. This vent field is called Lost City. Maybe you have heard about it in the past. There have been countless papers about Lost City looking at the origin of life at these sites. And it's a contender amongst others of probable sites that may have contributed to this. But really for relevance to this talk, the reason I bring this up is because it evidences the incredibly long evolutionary histories that have occurred in the ocean, especially at this microbial level. And with these long evolutionary histories, we see incredible genetic diversity. This becomes important and relevant from a health context when we start looking about where we might find, where we should look for molecules and compounds with commercial, industrial, or health applications. Many of these might be referred to as secondary metabolites, or metabolites, small organic compounds produced by organisms that can have relevance to humanity. When we look at where these secondary metabolites have traditionally come from, we see many have come terrestrially. Part of that is due to access, and many have also come from the marine environment. Many of these metabolites, many of these molecules can have anti-cancer, antibacterial, antifungal, antiviral, pest resistance attributes or uses, and of course many more. When we look at the distribution of these coming from the marine environment, we notice that the box is smaller. And one reason why that might be is because of something that we like to call the uncultivated majority. When you're working in these extreme environments, the ocean itself entirely being considered one, mostly because of its extreme lack of nutrients, considered oligotrophic or extremely low nutrient environment, we oftentimes can't cultivate these organisms by putting them, for example, on an agar plate or in a nutrient broth. That level of nutrient actually is toxic to many of these organisms. And so the mere cultivation of these organisms to study their potential from a secondary metabolite perspective, from a health perspective, becomes challenging. Here's again one of the further limitations of being in such an applied space. We can see that over time, though, the number of cultured isolates has been increasing, so we're making progress. And there's thought that in the future, new cultivation techniques will help expand this marine circle and let us really utilize the full extent of the ocean's genetic potential. Indeed, here are some of the ways of visualization of some of the new cultivation techniques being employed in these applied settings. Anything from, for example, nutrient flow assays, where microbes essentially can choose exactly what nutrient environment across a fluidic gradient that they want to live in, because we can't exactly predict what their best ideal environment is. Engineering of existing cells that we know can function with genes or genetic elements from the environment such that we can study their function. And a whole nother list that I won't get too detailed on, but if you're curious about talking about any of these, I'd chat more. Indeed, this topic of marine natural products and marine genetic potential, as of even just 2022, was listed as a very new, exciting, and quickly evolving field of study. On top of that, people are becoming interested in these marine natural products, this biomimicry and its, or even just tapping into this genetic potential and this vast genetic reservoir. The EU, for example, created the Blue Bioeconomy Forum, which had clear targets to evaluate ocean systems for example, biotechnology, bioprospecting, whether that's genetic diversity, cultivation strategies, or novel pharmaceuticals. Unfortunately, when we look at genetic potential in the environment in the context of climate change, we can frame it in the context of these, for example, planetary boundaries. The IPCC lists these boundaries as essentially key thresholds where if we pass, we can expect challenges. We see that biosphere integrity in the top left includes genetic diversity. And this genetic diversity has already been exceeded into the high-risk region. In this context, genetic diversity means genetic diversity loss or biodiversity loss as a result from climate change, warming conditions, changes to population structures and microbial competition, etc. Interestingly, when we look at these planetary boundaries, we also see that we've exceeded in the biogeochemical flows area, which connects to our discussion on global biogeochemistry from before. One last topic that we haven't quite addressed yet looks in this region at novel entities as a result of our changing environment. You can see that there's very little global quantification of this. When we look at novel entities or environmental pathogens, for example, we can think about drivers that impact their distribution, their severity, and their modes of infection. Here's a list of some of them, for example, changes in land use, or for example, water contamination, population health, travel even, and climate change you can see down at the bottom. But if we really think about these, we can actually say that a significant number of them, if not maybe all of them, are actually at least partially mediated by environmental factors. When we actually go to map how these parameters impact infectious disease more broadly, that becomes incredibly challenging and incredibly convoluted. We again see topics we've discussed today, like for example, increased temperatures. We see extreme weather events. But actually getting from those perturbations down to their impact in the context of, for example, airborne, waterborne, vector-borne diseases is still challenging and an active field of research. What we do know from looking at disease trends is that over half of our known human pathogenic diseases are aggravated by climate impacts. Here's a recent study in 2022 that evaluated this. And you can see, worryingly in the top left, 78% of these diseases are exclusively aggravated by climate-related issues. An example of this came from another study, for example, in the US that tracked correlations between El Nino-driven precipitation anomalies with infectious disease trends. In the US, this study found associations with hantavirus and plague. In Tanzania, they found associations with cholera. And in Brazil and Southeast Asia, they found associations with dengue fever. So you can see that these interactions are important to look at, yet still undercharacterized. So looking at the bigger picture, when we look at these vast web of interactions, we can see that these interactions exist even if they're nonlinear. I don't expect you to really take too much from this figure other than the fact that these connections exist, but we might need to look a little bit deeper beyond their direct, immediate connections to really evaluate them, especially since so many of these connections occur via an intermediate, like, for example, a disease vector or airborne transport. What we do know is that these impacts are real and that we should be focusing on them. And so when we go back to this initial question that we started with, why think about the ocean, or maybe more broadly, why think about the environment in the context of health, hopefully this list that we've generated here, that we've talked about today, can start to get your minds thinking about how it might connect to your work. But I really hope what I can leave you with is a willingness or a desire to evaluate some of these maybe non-obvious connections between the research you're doing, or, for example, the interventions, the climate interventions that you're pursuing, and the impact that those might have, both positive and negative, in the context of the environment. And so with that, I'd love to open it up to questions. Thank you for listening.
About the pH levels, acidity of changing?
Okay, about it? Yeah. So the question was, what about what we hear so often about ocean acidification, changes to ocean pH levels? We talked about it a little bit, but not explicitly. Really what it relates to are those changes in carbon speciation, mostly driven in part by biological cycling, but also by those acid-base reactions that are occurring in the ocean. The ocean, by its very nature, is a buffer. It has a lot of elements and compounds that naturally buffer the system. We've actually made use of that. It's one of the reasons why it's such a good sink of carbon, especially the excess carbon that we've emitted since the Industrial Revolution. Though the ocean can't quite keep up. And by inputting all of this carbon, we are changing the speciation, which shifts, again, it's an acid-based chemistry, it shifts the pH of the entire system to be more acidic. The impact of this is relatively severe for certain organism types, especially the organisms that form calcium carbonate or carbonate-based structures. This can be, for example, shells, this can be corals, and a whole slew of other, for example, even microbial organisms, like I believe coccolithophores, that form calcium carbonate coccoliths over their structures. So it has wide-ranging impacts to the essentially survivability of key organisms across our entire trophic ladder. Many organisms are quite resistant to this acidification, but interventions to hopefully counteract acidification is a big field of research right now. Indeed, there's a compound that was recently produced out of some chemical oceanography professors at the University of Washington that's shown to potentially interact with some of these inorganic carbon species once captured or once mixed, and can actually, I believe the mechanism is precipitate many of these carbonate species out of the seawater, essentially purifying it from the excess carbon that we've emitted. And I believe some of those byproducts can actually be used for biomaterials. And so there are some really interesting interventions that target this problem and hopefully try to solve it.
Other questions?
Hi, Dylan. It's nice presentation.
Thank you.
And my question is, you are talking about like genetic modification based on the recent changes, climate. So how the microplastic may influence the genetic modification inside the species of the ocean?
Yeah.
Have you ever thought about that one, or in any studies is ongoing like that?
Absolutely. So the question was, how do microplastics impact a whole slew of these issues that we've talked about today? Directly, it's quite unknown. Microplastics, and maybe I'll expand the question slightly to talk also about persistent and emerging organic compounds that we are also emitting out into the environment. There are all of these forms of new, novel, and highly unknown and undercharacterized pollutants that oftentimes are bioactive. In the context of, for example, emerging and organic pollutants, that might take the form of drugs that we flush down the toilet or that don't make it through wastewater treatment and end up in our natural environment. There are quite a few studies that show that those compounds interact adversely with populations in the environment across a whole slew of trophic levels, oftentimes with the most noticeable impacts at the higher trophic levels, which can impact, for example, our fisheries and our malnutrition. At the microbial level, and with plastics specifically, these interactions are significantly less known. I believe some recent work found some plastic degrading bacteria, which is really interesting. More so, what plastics can allow for is essentially a particulate environment for organisms to grow. We're creating all of these microparticles where bacterial settlement can create these microenvironments that can lead to a whole slew of sort of localized genetic transfer, localized evolution. What exact impact that might have, I think, is still heavily under research, but thank you for the question.
Sort of on the subject of carbon sequestration, I know that there's investigations into using algae as carbon sequestration technique, but I don't know if anybody's investigated just shellfish farming as its own carbon sequestration technique. So I wanted to know if you knew about the relative efficacies of those two possible techniques.
Yeah, absolutely. So the question was essentially two major ideas for carbon sequestration using algae, using potentially shellfish or fisheries. And there are also many more. I mean, there are also the form and the way in which these interventions are carried out also vary widely. So, for example, I mentioned algae growth through farming and then also use as a micronutrient supplement, or, for example, replacing some of our traditional foliage with algae-based foliage. There are also ideas to grow a bunch of algae and then sink it into the deep sea as sequestration, so bypassing the entire sort of metabolism, at least human metabolism and regeneration issue that we had talked about. Algae specifically, which I know a little bit more about, is really an incredible organism. I believe every year they have the highest flux of carbon of any living organism. The rate of carbon fixation is incredibly high, and they cycle a lot of carbon every year. I think it's a very promising strategy. I think really the perspective we need to take with all of these different strategies is to look more deeply at their impact. And I think one way in which we do that is by keeping in mind this idea of residence time. What impact will these strategies have, and especially on what timescales do they operate? Perhaps maybe sinking, for example, maybe kelp is the solution, but sinking kelp sequesters carbon for a longer period of time than the others do. The other thing to keep in mind are the unintended outcomes. For example, what if we sink kelp and it contributes to respiration in the water column as organisms start to degrade this freshly introduced organic matter, and that actually leads to a huge flux of carbon into our system that we didn't originally anticipate. So it's a complex system, it's a complex web, and unfortunately that means usually answers are complicated and we're still learning. One other thing related to that might be, for example, iron fertilization. Maybe you've heard about that. We're dumping a bunch of iron, or essentially limiting nutrients in the environment, can stimulate primary productivity, draw down carbon. There's a famous quote by one of the early proponents of iron fertilization that says, "Give me a tanker load of iron and I'll give We're now seeing that the system is maybe more complicated than that. There are more complex nutrient limitation going on in different regions of the ocean, and also the very cycling we looked at today might actually make the sequestration of that organic matter, how much of it actually gets exported out of our ocean atmosphere system, more unknown.
Yeah, so, yeah, great. Very interesting talk. Kind of
Thank you.
just, so 2 questions. So my first question regarding to the nanopore sequencing part. So because there's a lot of unknown species, right? Kind of stuff from, I guess, especially from the deep ocean, deep area or different extreme kind of environment. So did you guys have any kind of challenges to deconvolute those kind of sequencing and just like with the nanopore?
Absolutely.
Yeah. So
yeah, it's a great question. More specifically, I think a lot of these next generation long read sequencers, and nanopore specifically, has made significant jumps in terms of their quality and reliability of their actual sequencing. For a long time, we actually felt like we couldn't use nanopore because of the quality of the sequence output, or really the reputability of the sequence output. The other thing you have to take into account is that the coverage of our sample, being a metagenome, is oftentimes a lot lower. And so we have a lot less agreement, essentially sequence agreement, to go on when we're making these distinctions. Now that Nanopore has released their Q20 chemistry, we feel more confident in using those techniques. We think that the sequencing error rate is low enough that we can produce sequence result that's compatible or comparable to something maybe like a PacBio Hi-Fi, or especially sometimes the Illumina short reads. But we also think that the benefit of having these really accessible long read sequencers, and also the benefit of having these long reads when it comes to, for example, reassembling extremely genetically diverse, for example, viruses with a lot of genetic diversity, genomic rearrangement, having these long reads ends up being really important. And so we've kind of gravitated from that perspective to the PacBio Hi-Fi nanopore world. And we chose nanopore because we think that the Q20 chemistry brings it to a level that's comparable.
Great. My second question is kind of regarding, I don't know whether you noticed that recently there's a paper on Nature. They mentioned that they found some kind of called a dark oxygen. So which kind of those oxygen can be produced without light. You don't need any kind of plants to produce those oxygen.
Yes.
So how do you think those kind of oxygen contribute to the climate change, or just like the ocean?
That's a great question. I believe there are I would call 2 types of dark oxygen. The first that I believe this Nature paper is referring to is oxygen production at, for example, deep sea polymetallic nodules, or these sort of manganese heavy metal nodules that we see in certain deep sea environments. There is a lot of complex chemistry, geochemistry, biochemistry going on in the ocean, and they're all interacting. My specific focus isn't as much on the nodules. I believe the idea is that there's some kind of redox chemistry stimulated by this high density of heavy metals that's causing essentially maybe a breakdown of the water producing the oxygen. I'm not entirely sure on that though. Otherwise, the other kind of dark form of energy of oxygen that we might see in the environment would be something that we would call cryptic oxygen cycling. Cryptic specifically refers to the fact that whatever is produced is immediately consumed. And so we see these apparent 0 oxygen concentration levels, like for example, in the oxygen deficient zones that I mentioned earlier, when actually there is an oxygen cycle occurring. It's just immediately being consumed, and oftentimes the life that's consuming it might be like oxygen limited. And so I think both of those scenarios in a changing environment where maybe, for example, we'll have increased organisms producing oxygen in these oxygen deficient zones, it makes the question of how these might evolve in the context of climate change very uncertain, or how these might evolve in the context of, for example, polymetallic deep sea mining, very uncertain. Yeah. Mm-hmm. I think really what the nodules are dense. They're formed by unique geologic processes, and they're dense metals that are occurring at the ocean sediment interface. So they're sitting on the bottom. They host really interesting microbial metabolisms, but they're less related to the actual fish in the area. The fish, though, would be a great example of what might be contributing to a cryptic oxygen cycling in that area, where any oxygen that's produced is probably pretty immediately respired. Online question. Let's see. Yeah. Oop. Here we go. Oh, I don't think I can see the main webinar chat.
Okay, let me read it. So the question is, hello? Yeah, the question is the importance of diatoms in the ocean is ecosystem and the interaction with the atmosphere. Is microplastics affecting the diatom?
It's a great question. So it's specifically asking the importance of diatoms, which are a small, they're small photosynthesizing organisms in the ocean. These diatoms, importantly, produce a lot of oxygen for the world. They engage in a lot of photosynthesis and they fix a lot of carbon. And they act as a foundational member of the global food web, and especially the marine food web. The question was specifically, are microplastics impacting diatoms and thus the broader system? And I think the answer is it's still highly unknown. That being said, a lot of the regions where diatoms occur, especially in the open ocean, in the oligotrophic gyres, are also the regions where, for example, you may have heard of the garbage patch, where a lot of these microplastics are occurring. What we do know is that a lot of these pollutants, both plastics and these emerging and persistent organic compounds, are bioactive, do tend to be bioactive, and so they very well might have an impact on the system in ways that we might not currently know.
Another question is, are these dead zones due to soil erosion and fertilizers of nitrogen and phosphorus that causes algae bloom that consume the oxygen within that zone?
Yeah, so the question was specifically looking at the oxygen deficient zones. Are these zones driven by anthropogenic pollution like fertilizers or soil erosion? And I think the short answer is those might be contributing to more localized, what we might call anoxic regions or anoxic zones. Oftentimes the input of these nutrients causes a boom in respiration, which depletes the water column of oxygen. This happens in more localized areas. For example, you might think of some marinas that lead to fish kills we see in the news around Southern California, for example. The broader ocean system, though, is large enough where these processes are mostly naturally mediated, but they operate in a very similar mechanism where nutrient input and stratification in the surface, and especially sinking nutrients by primary productivity, in the surface ocean, leads to remineralization and respiration in the deeper water column, but without access to oxygen transfer from the surface ocean leads to these broad dead zones. So it's a similar concept and a similar mechanism, but I would say on a larger scale, and it has a natural mechanism.
Thank you. Final question is, since climate change causes ocean stratification, can we design a thermoelectric generator to protect the coral and also reduce the ocean surface temperature in the tropics near land to see if this might weaken hurricanes as it approaches land?
Yeah, absolutely. So this question was pertaining to essentially the efficacy of one of the climate interventions that's a bit more technological. The idea is to essentially create some kind of structure that could cool the surface ocean. Oftentimes that's done by pumping deep ocean water up into the surface. There are a few different proposed mechanisms by which this can be done. As you can imagine, it would require a lot of energy. And so one way in which people have proposed to do it is to utilize, for example, tidal fluxes, tidal energy, perhaps even wave energy, to pump this colder water up to the surface, counteracting the effects of climate change. I think it's an interesting idea and an interesting solution. I think we have to be aware of the broader changes we might be causing by pumping not only colder water, but nutrient-rich water back up into the surface ocean. We might be inadvertently causing more issues than we might be solving with some of these solutions. And we have to keep in mind the energetic demand to do this Perhaps there are ways to exploit natural systems like tidal and wave energy in order to create this pumping, but the scale that we have to operate in order to counteract the effects of these global cycles might be unfeasible, at least for now. But it's absolutely being discussed, and this solution is proposed and out there.
Thank you. Final question. Have you associated prehistoric mass biodiversity loss to phytoplankton or diatom loss?
Yeah, absolutely. So it's looking more on Earth's geologic history. The question was asking, can we essentially use our past mass extinction events or biodiversity loss to better understand what's happening now? And the answer is absolutely. If we look at one example of past biodiversity loss, one key time point in Earth's history might be at this Paleocene-Eocene thermal maximum, it's called, which is the transition between the Paleocene and Eocene. And at that thermal maximum, we saw extremely high average surface temperatures, and we saw one of the largest biomass collapses and mass extinction events. When we compare that time frame to now, or some of these past mass extinction events to now, what we actually see is that modern day biodiversity loss and modern day temperature change actually outpaces all of our historical naturally occurring climate-related biodiversity loss. And so that rapid change of average global temperatures and average global conditions is what we're really worried about. And it suggests that maybe we can expect worse conditions than in some of these older and to some degree slower global changes.
Thank you. That's all I have.
Great. Thank you, everyone. Sorry to keep you late. Thanks. Thank you.